Every quantitative systems pharmacology model is, at its heart, a map. A map from a parameter space, the set of all possible configurations of your model, to an output space, the curves you actually measure in the lab or the clinic. On the left, picture a plane. Two axes. Two parameters, θ1 and θ2. Every point in the plane is one possible configuration of the model. On the right, picture an output axis. Concentration over time. Each configuration, pushed through the model, produces one curve. Here is the uncomfortable geometric fact. Sometimes, two completely different points in the parameter plane produce exactly the same output curve. You can watch the system forever, and the data will never tell them apart. This is structural non-identifiability. Geometrically, it isn't two isolated confusing points, it is an entire line, or even a surface, in the parameter space, where every single point on it maps to the same trajectory. And here is the good news. The fix is always another measurement. When you add a second observable, a new marker, a new tissue readout, a second output axis appears. The non-identifiable line collapses down to a single point. The configurations that looked identical from one angle are now distinguishable from two. Structural identifiability analysis tells you, before you ever touch real data, which parameters are trapped inside non-identifiable manifolds. And it tells you exactly which measurement will break them apart. The next geometric question. Where in this vast parameter space do biologically plausible solutions actually live? Picture the prior as a three-dimensional cube. Every point inside is a priori possible, the full range of each parameter from literature. Now apply biological constraints, one at a time. Is the growth rate positive? That removes a slab. Does the system settle to a stable steady state? Another slab. Are the immune proportions within the observed range? Another. After every constraint has sliced through the cube, what remains is almost never a nice big ball. It is a thin, curved sliver. A warped sheet, occupying a tiny fraction of the original volume. And it isn't round, it twists. Now imagine scattering random samples into the cube. Most miss the sliver entirely. They land in gray regions, in feasible configurations. Only a few by luck land on the thin feasible surface. Suppose you draw 100 samples at random and only 3 land inside. This is the geometric picture behind a common, painful reality in QSP modeling. If the feasible region is this thin, random initialization for Bayesian calibration will systematically fail. The solution is Latin hypercube sampling, a stratified design that spreads your samples evenly across every projection of the prior. Even with the same sample count, LHS hits the feasible sliver at a higher rate than random draws, because it can't cluster in any single region. Geometrically, LHS gives you good coverage with relatively few points, enough to see the shape of a high-dimensional region. Calibration asks a geometric question. Given the data we observed, which parameter configurations remain plausible? The answer is a cloud, a posterior distribution over parameter space. Here is how it is built, geometrically. Start with a broad scattering of particles across the prior. Round 1. Draw a fat, translucent shell around the region of parameter space that reproduces the observed data within a loose tolerance. This shell is wide, many particles survive. Contract the shell. Round 2, tighter tolerance. Fewer particles survive. The survivors resample around their neighbors, creating a denser cloud in the surviving region. Contract again. Round 3. The shell is tight now. The particles that remain form a compact cloud. That cloud is the posterior. Zoom in. This isn't a single point estimate. It is a distribution of points, and the shape of the cloud tells you which combinations of parameters are mutually consistent with the data, and which aren't. Narrow in one direction means you've pinned a parameter down. Elongated in another means the data leaves that direction loose. The geometric intuition is simple. A calibrated model is not a pinpoint on a map. It is a small, well-shaped region. Structural identifiability asked whether any data could, in principle, resolve a parameter. Profile likelihood asks the practical question. Does the data we actually have resolve it? Picture the likelihood as a landscape. Mountains and valleys over the parameter plane. The tallest peak sits at the best fit configuration. Now, for each value of θ1, look along the θ2 direction and find the highest point on the landscape. That highest point is what the profile records. Sweep across θ1, and the string of these peak heights forms the profile curve. In effect, you are tracing the ridgeline of the landscape, projected down onto the θ1 axis. The shape of the profile is the identifiability verdict. And it comes in 3 archetypes. Archetype 1. A rounded peak in the landscape. The ridge sharply descends on either side, so the profile is a sharp single peak. The data has pinned the parameter down. This is an identifiable parameter. Archetype 2. A long ridge in the landscape, extending in the θ1 direction. At every θ1, the maximum over θ2 is roughly the same. The profile is flat. No matter what value θ1 takes, you can compensate by adjusting θ2 and still fit the data just as well. The parameter is practically non-identifiable. Archetype 3. A cliff, a peak with one side dropping off steeply, but the other side extending as a plateau. The profile has a sharp wall on one side and a flat plateau on the other. Partially identifiable. The data sets a lower bound but not an upper one, or vice versa. Structural identifiability asks, could any data do this? Profile likelihood asks, does ours? The 5th question is fundamentally different. It isn't about parameter space. It is about how the system behaves in time. Picture a phase portrait. 2 axes, 2 concentrations, x1 and x2. Every trajectory the system can trace through time is a curve on this plane. When the loop gain is below 1, subcritical, the flow looks like water draining to a single point. Every starting condition, no matter where, spirals inward toward the origin. The cascade decays. The system is quiet. Now turn a dial. The loop gain increases. At exactly 1, the spiraling stops. Neither inward nor outward, the damping vanishes, and trajectories drift on neutral circles. The system is perfectly balanced on the threshold. Above 1, supercritical, the balance breaks in the other direction. Starting point spiral outward toward a stable closed orbit. The system self-sustains. It runs forever. This is a bifurcation. And here is the geometric punchline. A bifurcation isn't a number crossing a threshold. It is the entire shape of the flow reversing direction at once. Side by side, 2 phase portraits. One below the cliff, where every trajectory dies. One above, where every trajectory persists. Between them, no smooth transition, only a qualitative flip. This is why some mistunings don't just slow a system down. They change whether the system runs at all. The 6th question. Which parameters drive the output most? There are 2 ways to answer it, and the difference between them is entirely geometric. Picture the output as a surface over the parameter plane. A rolling terrain. Height equals output value. 1 at a time sensitivity, or OAT. Pick a point, the calibrated configuration. Walk a thin strip along the θ1 axis, holding θ2 fixed. The height range along that strip is the OAT swing for θ1. Do the same along θ2. Rank the swings in a tornado chart. OAT geometry is local. It tells you which parameter matters at this spot. Sobol sensitivity asks a global question. Instead of one strip, scan the entire surface. Decompose the total height variance into 3 parts. First, the variance explained by θ1 alone, how much the surface tilts along one axis averaged over the other. Second, the variance explained by θ2 alone. Third, critically, the variance from their interaction. The part of the variance that only emerges when both parameters move together. These three quantities stack into a total variance bar. And here is the geometric difference OAT cannot see. If two parameters only matter when they vary together, if the surface has a diagonal ridge or a saddle, OAT's thin strips miss that structure entirely. Sobol captures it. OAT tells you which parameter matters at this spot. Sobol tells you which parameter matters across the whole landscape, including ways they only matter together. The seventh question closes the loop. How do we turn a posterior in parameter space into a prediction we can trust, and where are its limits? Start with the posterior cloud from calibration, sitting on the parameter plane on the left. Draw the model as an arrow pointing right. Now carry each particle along that arrow. It lands somewhere on the output axis. Do this for every particle in the cloud. A histogram builds up on the output side. This histogram is the push-forward, the posterior distribution pushed through the model. The middle 95% of the histogram is the 95% credible interval on the prediction. A credible interval is not a mysterious thing. It is a picture. It is the shape you get when you take a cloud in parameter space and carry it through the model. And now the coda. Focus on a single particle. Perturb its parameter along one direction, and watch what happens to where it lands. At first, the output moves. As you push the parameter further, the output keeps moving. Then something changes. The output stops responding. You keep turning the parameter dial, and the landing point on the output axis refuses to budge. You have reached the ceiling. Geometrically, the ceiling is where the push-forward map flattens. The input is still changing, but the output has hit a wall. There are two reasons this happens in biology. Saturation, a receptor, an enzyme, a feedback loop is already at maximum. Or structural inaccessibility, the parameter you are turning controls a pathway that doesn't actually reach the endpoint you are measuring. Either way, the lesson is the same. If the push-forward map has flattened, more drug won't help. More dose won't help. The ceiling is biological, not pharmacological. 7 questions. 7 pictures. Every picture shows you something an equation alone never could, the shape of what your data can and cannot tell you.